Okay, we're going to continue our discussion in introductory biology on biomolecules and biochemistry by starting to look at one of the really important classes of biomolecules, which is carbohydrates. A lot of people have heard of carbohydrates in a dietary context, but we're going to look at some of the underlying chemistry and then start to look at the biological function of these really important molecules. So we'll start off just with the empirical chemical formula of carbohydrates. So carbohydrates all have the formula which is there, which is CnH2nOn. So what that essentially means is that for every carbon, C, in the molecule, there is an equivalent number of oxygen and twice the number of hydrogen atoms. So you can look at this or remember this by just looking at the name. So carbos is in reference to carbon, hydrates is in reference to water. As we can see, for every carbon there is an oxygen and two hydrogens. The empirical formula for the molecular formula for water is H2O. So by looking at that, we can see that for every carbon there is an equivalent water molecule. So we can just essentially look at this as carbon and then water. So we have hydrated carbon, or carbohydrates, essentially. So we have a few examples of this in what are called monosaccharides, or just singular units of sugars. We'll get back to this monosaccharide, disaccharide, polysaccharide idea in a second, but some of the main common examples you'll see are, first off, we have glucose. So glucose is one of them, is pretty much the sweet thing that you eat and your body takes and metabolizes to make energy. So glucose has the formula C6, so 6 carbon atoms, H12, 12 hydrogen atoms, O6, 6 oxygen atoms, and you can see it follows that empirical formula. And it has one structure, but it can be in multiple configurations. So it has both this straight chain structure here and this cyclic structure here. In water, when glucose is in water, the straight chain structure and the cyclic structure are in equilibrium. So there is a constant proportion of straight chain to cyclic, and they're constantly interconverting. So yeah, they exist in an equilibrium in solution. Another example of a sugar is this ribose sugar here, C5H10O5, and it's quite an important sugar as well. Glucose is important for energy and thinking about where cells get their energy, but ribose is also very important. Here's the cyclic form. It also has a straight chain form, but the cyclic form is more pertinent because it has an important role in RNA and DNA, the polynucleotides that make up our genetic information. So in RNA we have this form, ribose, which forms the R of the RNA, which stands for ribonucleic acid. We'll come back to this all in later lectures, but it's important to note that a carbohydrate is part of these larger biomolecules, and ribose is quite an important one to look at. In DNA, you have the D stands for deoxyribose, so essentially you have this ribose, and then you don't have this oxygen here, and it's deoxy, essentially. So this oxygen here is gone. Anyway, with naming carbohydrates, they're generally named with how many carbons they have. So they're named with the sort of prefix of coming from the Latin words for number of carbons. So you have one carbon, one mono, two di. You don't really see those in carbohydrates there, but 3 is tri. 5, the common ones you see are 3, tri, 5, pent, 6, hex, and the root to identify a carbohydrate is ose. So for example, glucose here, it has 6 carbons, so it is hex for 6, and ose, it's a hexose sugar, 6 carbon sugar. Whereas ribose here is a 5 carbon sugar, and so it is a pentose sugar. So that's just generic naming of sugars based on how many carbons they have. So what we're going to look at now is what happens when you build up these sugar units into these multiple small units into what are called di and polysaccharides. So disaccharides are when two carbohydrate units, so something like glucose, one singular unit, are linked by a glycosidic bond between them. So a glycosidic bond is just a bond where essentially you have a reaction between these two hydroxyl groups on the sugars. So these two groups react, right? And what happens is you end up so if you looked at it in sort of macro, you'd have an oxygen and a hydrogen here, and an oxygen and a hydrogen here, and this oxygen attacks this hydrogen here, and this bond forms between here, and these go off. So essentially what you have is you have a rearrangement between these two hydroxyl groups where a water molecule is spit out, and you get this linkage through one oxygen atom in what's called a glycosidic bond. So in our example here we have glucose and we have what's called galactose, which is an isomer of glucose, so it's structurally very similar, and in fact its chemical formula is exactly the same. And when they're linked together in this glycosidic bond, so where you take out a water molecule between them and form this linkage through this oxygen atom, you get what's called lactose, and lactose is the disaccharide you find in dairy products like milk. So disaccharides are small, there's many examples, and they're generally found for energy storage and energy shuttles, so moving sugars from one place to the other. They're very useful for that. If you do this multiple more times, you get what are called polysaccharides. So polysaccharides are multiple carbohydrate units, like into the hundreds, all linked together in the same way. So if you constantly just build up this chain of units, and by linking them all in the same way, by forming these glycosidic bonds between all of them, you form very long chains of carbohydrates, or very large polysaccharides. And examples of this are starch, which is found in things like potatoes, so that's used for energy storage in those sort of vegetables, and things like cellulose, which make up the cell wall of plant cells, which we'll look at in more depth when we get to cellular structure. But cellulose is an important structural molecule to help support the plant cell and help it keep its structure, and it's a huge chain of these carbohydrate units. So we're just gonna, that's some of the underlying chemistry, looking at the general structure of carbohydrates, some of the naming, and looking at di and polysaccharides. So we'll just look at some of the roles carbohydrates play. So the first is energy storage. So many polysaccharides, because they are just multiple units of these monosaccharides, which are highly energy rich, they're used for energy storage. For example, starch in plants, as we mentioned, in potatoes, that's a form of energy storage for the plant. And glycogen is our form in animals and humans, is sort of our form of starch, and they're very similar to each other. And they're both polysaccharides and highly important for energy storage. Carbohydrates are also very important as an energy source. So sucrose, which most people would know as table sugar, is a disaccharide of glucose and fructose, and so it's just glucose, which you've seen, and a different carbohydrate named fructose, which is structurally, it's a pentose sugar, structurally quite similar. They together have a glycosidic link between them in a disaccharide, and many animals, including us, eat that for energy. Another example, which we saw before, was lactose, which is, as we saw, a disaccharide of glucose and galactose. It's in milk and dairy, and it's an important energy source for many animals, particularly young animals who drink their mother's milk. As we also discussed briefly, carbohydrates play an important role in structural roles. So cellulose in plant cell walls is a polysaccharide of glucose, and it forms extremely strong chains and fibers which are able to give plants its strength and build things like trees and tree trunks, which are mostly cellulose. One of the funny things is we can't metabolize cellulose, but we can metabolize starch, and they are essentially almost the same chemical. Cellulose is a polymer of what's called beta glucose, which is an isomer of the glucose cyclic unit. There's two isomers, there's alpha and beta. Cellulose is a polymer of the beta units, and because of our biochemistry and our proteins and how specific they are, we can't actually metabolize this, and no animals can. All the animals that eat plant matter need symbiotic relationships with other organisms, whereas starch is a polymer of alpha glucose, which fits perfectly with our biochemistry, so we can easily metabolize this. So it's just interesting looking at how small changes in chemistry can have dramatic effects in what we can and can't metabolize, and dramatic effects on the function of these relatively similar molecules. Anyway, that's it for this video. Next video will be on lipids.